Okay, welcome to another screencast about cellular respiration. I remember as a student learning this and not comprehending why I should care or why it matters. And then once I started teaching it, I started to all the dots kind of came together, and I started to really see how and why this is such an important topic to learn about. So I encourage you as we go through this to try to relate it to your real life, right? When we're talking about fermentation, think about exercise and lactic acid buildup and sore muscles. When we're talking about cellular respiration, think of oxygen consumption and breathing out CO2 as waste, and the food that we eat and how that is converted into energy. And then the topic becomes really cool, I think. All right, so I'm going to share my screen and we will go on to my PowerPoint slides. Okay, and let me fiddle around with this really quick so I can see multiple things on my view, but hopefully you can just see the shared screen. Okay, so we are talking about cellular respiration, which is really harvesting chemical energy from our food, right? And again, the chemical energy that we are harvesting is primarily from C6H12O6, which is glucose. But I'll show you at the end how we can also harvest energy from foods like fats, other sugars, proteins, etc. Okay, can you close that door all the way, please? Okay, so we've already kind of talked about how photosynthesis takes the energy from the sun, and it harvests that energy. And we're going to talk about that in the next section, how it does that through binding of chlorophyll, which is a pigment. And so photosynthesis takes sunlight, water, and carbon dioxide and converts that to C6H12O6, right? So that again is anabolic because you're building it up. It's endergonic because we need the energy from the sun. And we're powering that up to produce glucose. And plants give off oxygen as their waste. In cellular respiration, the topic that we're going to talk about today, we are breaking down glucose, right? We're going to see waste as CO2. We're going to need that oxygen. We're going to get water out. And in the process, we get a lot of ATP, right? Energy. Right? The bonds in ATP are formed. And therefore, this reaction is because we're breaking something down. It's going to be catabolic. It's exergonic because we get the energy out in the bonds formed with ATP. And we get that overall process, right? Where we release CO2 as waste. So just a reminder of those two. It's really important to mention that the sun on this planet is the entire biome, the whole biosphere, the Earth. It's our main source of energy. All living cells require energy from an outside source. And the sun provides that outside source to all of our cells, right? So plants use energy from the sun, right? And they use that to convert to C6H12O6, which they can then break down to give themselves ATP. We, in animal cells, we eat plants, or we eat other organisms that eat plants, right? So plants really are the end-all be-all as a way of capturing that energy from the sun. So photosynthesis is used to, again, make organic compounds. So again, those organic compounds primarily are glucose, and they make oxygen, right? Whereas cellular respiration uses the chemical energy from glucose to create ATP, and in the process, it gives off carbon dioxide and water. So cellular respiration can really be defined as the catabolic pathways. Remember, catabolic means breakdown, right? The breakdown pathways of aerobic and anaerobic respiration, which will break down the organic molecules to make ATP. So aerobic means with oxygen, and anaerobic means without oxygen, right? So fermentation, so we have these two processes. So this is without oxygen, and then we have aerobic that is with oxygen. Whoops, my bad. So we're gonna mention both of these. So fermentation and anaerobic respiration requires no oxygen. This occurs in the cytoplasm, meaning that these are very ancient processes. So evolutionarily, before organisms, before cells went through kind of that endosymbiont process to create mitochondria and chloroplasts, they had just the cytoplasm. So these processes could create energy by just their cytoplasm. However, because of that, they have a very limited amount of energy, so they don't get a lot of ATP out. On the other hand, aerobic respiration requires oxygen, right? It occurs, sorry, I had to close my window because my son is loud. There we go, requires oxygen. It occurs in the mitochondria. So that means it's only eukaryotic cells that we're dealing with here, whereas fermentation and anaerobic are all cells versus only eukaryotic cells. And in aerobic respiration, we see the release of very large amounts of energy, all right? We get a lot of ATP made. Sorry, I'm trying to, there we go. Close up my little, trying to get a better view on my screen. Okay. So if we look at this, the anaerobic, here, I'm gonna change that, make it red. So the anaerobic processes are occurring in the cytoplasm. I hate spelling that word, aerobic. Anyway, and yeah, and I spelled it wrong, whatever. So anaerobic are in the cytoplasm. These are basically all cells go through the process of glycolysis, right? So it's a very ancient process. Whereas the aerobic respiration occurs in the mitochondria and therefore only occurs in eukaryotic cells. But that has allowed eukaryotic cells to get much bigger and expend more energy because we get more ATP out, right? Mr. Anderson has some great videos, which I have linked online as well. All right, so cellular respiration is a series of redox reactions, all right? And I know some of you are like, ooh, redox, chemistry, yay. And other people are like, redox, chemistry, ah. But we're gonna make it very simple, all right? So redox just means oxidation and reduction reactions. And hopefully you learned one of these silly acronyms in chemistry. Oil rig, anyone? Oil rig means oxidation. Oxidation is loss. And here we're talking about electrons, right? And rig, meaning reduction, is a gain of electrons. Another one that I learned in college that I really liked was Leo says Gur. So loss of electrons is oxidation, gain of electrons is reduction. So however you want to remember it, but you do need to remember it. But this is really confusing for some people. So if we look at it, oxidation is a loss of an electron. And what that means is it's a decrease in energy, right? So in biology, in chemistry, we're always talking about electrons. In biology, it's easier to think about the hydrogens, all right? So in biology, I like to think of it as a loss of a hydrogen, right? And the way that I remember that is the oxidation has an X in it, and so you're going to remove a hydrogen, right? So reduction is a gain of an electron. You get a increase of energy because those electrons have energy associated with them. And in biology, we're often talking about a gain of an H, right? So oxidation, you are Xing out an H, and in reduction, you are gaining an H, all right? So if we look at the reaction here, down here, for cellular respiration, we have glucose plus oxygen is going to water and CO2. So what is actually being oxidized here, right? And what is being reduced? So I think the best, easiest way to look at this is look at the carbons. We're going to be following carbons a lot through this process. Yeah, thank you. So if we look at carbon, where did it go, right? So so the glucose in the carbon in the glucose is actually going to this carbon here in the carbon dioxide, right? So what happened to the H's there, right? What happened to the glucose? So glucose, did it gain an H or lose an H, right? So it lost an H, meaning we're going to cross one of those H's out, meaning that it was oxidized, right? So glucose is oxidized to carbon dioxide. And if we look at now the O, what happened to this oxygen here, right? So this oxygen is going into the water and it is gaining an H, right? So oxygen was reduced. I know it's very weird. It gained something, but it was reduced, right? So oxygen was reduced to water, right? And you should definitely be able to look at a reaction and see what was oxidized and what was reduced. Another topic that we mentioned, we kind of touched on in the last part, was about coenzymes, right? So we know coenzymes help enzymes work better. So do cofactors. And coenzymes are more organic. So in cellular respiration, we're going to talk about two coenzymes that are really important for the process, and they act as something very specific in cellular respiration, right? So coenzymes in cellular respiration are going to accept electrons as glucose is being reduced. And so another way of saying that is that they are going to be electron carriers. That is kind of how the book and biologists talk about this, is that these coenzymes are going to be electron carriers as glucose breaks down, right? So if we look at our reaction here, and my arrows don't transfer there, that's why I get these little weird boxes. So here we have glucose is oxidized, right? So again, glucose lost an H, so it was oxidized to carbon dioxide. And as it does that, the coenzyme that the coenzymes here are going to accept those electrons that were lost, right? And they're going to carry them. So the two electron carriers that we're going to talk about are NADH and FADH2. And they have crazy names that you'll never have to know, but I'm going to tell you them. The NADH is called nicotinamide adenine dinucleotide. And it's a derivative of the vitamin niacin. And then the FADH2 is flavin adenine dinucleotide. You don't have to know so much about them. So if we look at NADH here, it is, so what this means is that electrons from organic compounds are going to be transferred to NAD plus to make NADH, right? And so going from NAD plus to NADH, is that a reduction or an oxidation, right? So we gained an H, so that is going to be a reduction. So this was reduced, right? Meaning it gained electrons. It gained an H and it gained electrons, right? And in fact, if we wanted to do the math, I don't really care so much, but it's gaining 2 electrons, right? So each NADH, every NADH made through cellular respiration is going to represent stored energy, and we're going to help use that to make ATP. So FADH2 is basically the same idea. We have electrons from organic compounds transferred to FAD. So we have FAD to FADH2, right? And just like NADH, this is reduced, meaning it gained a hydrogen and 2 electrons, right? So remember the NAD plus, you add one electron to make it NAD, add another electron to make it NADH. For FAD, to get it to 2 H's, we have to add 2 electrons. All right, so in an introduction to what is a really big part of the cellular respiration, which is called the electron transport chain. I will very often call it the ETC, right? Who down with ETC? Yeah, you know me. You guys probably don't remember that song. There was a song back when I was in 8th grade called OPP, Other People's Property. And yeah, that's what the ETC. Anyway, okay. Introduction to the electron transport chain. So here we're looking again at free energy diagrams. So we're looking at Gibbs free energy on the y-axis, and we don't really have an x-axis here. We're just looking at a flow down. So if you mix hydrogen gas and oxygen gas, this can be quite an explosive situation, right? You literally can, it's an explosive release of heat and light energy. And I think that you guys have probably heard of this through the Hindenburg explosion, the blimp that was filled with hydrogen gas. And then people were like, oh, that was a terrible idea. Because you get these uncontrolled, crazy reactions, right? It's an uncontrolled reaction. So what the electron transport chain does is that we're also doing hydrogen and oxygen. However, we are getting the hydrogen from our food, right? And that's going to be through NADH. And then we also have oxygen gas. And so it's kind of the same reaction as this crazy explosive reaction. However, 2 main differences. One, the H comes from organic molecules, not hydrogen gas. And the second is that there's a series of steps that will break the fall of those electrons. Rather than having this crazy explosion, we have very calculated steps where the electrons are moved from one molecule to another, one molecule to another, one molecule to another in a very stepwise fashion in order to create our ATP, right? To harness that energy rather than have an explosive reaction. So it releases energy gradually instead of all at once in this uncontrolled way. Okay, so cellular respiration has 3 stages, right? You have to know these. So the first stage is glycolysis, right? And if we remember the name, hydrolysis or hydrolysis was water cutting. Glycolysis or glycolysis is cutting of glucose, right? It's got a great name. So what we're gonna see is that we're gonna go from a 1, 2, 3, 4, 5, 6 carbon glucose. We are going to cut that, and the result is that we're gonna get 2 3 carbon sugars, and those are gonna be pyruvates, right? So glycolysis over here is that first step. It happens in the cytoplasm. It's very ancient. All organisms go through glycolysis. It's amazing. Our cells have the exact same enzymes as bacteria down in some crazy hydrothermal vent. It's amazing. Same enzymes doing the exact same process, right? It's really neat. And that generates a little bit of ATP, all right? So you get some ATP, but not very much. We're gonna see a net gain of 2 ATP through glycolysis. All right, so the next step is gonna be called the Krebs cycle or the citric acid cycle, right? And what this is gonna do is it's gonna break pyruvate down into carbon dioxide, right? So it is going to go from pyruvate to carbon dioxide. So we're breaking it down even further. And so when we look at the citric acid cycle or the Krebs, we're going to see that carbon dioxide is lost as waste. Oops. So citric acid cycle is here, and we'll see, again, we get a little ATP out, right? I think that's 2. I wish I had my notes with me. I think that's 2 ATP. Let's double check. course, I don't have it written down. I think that's 2, but we'll go over the whole drawings later on. So we get just a little bit of ATP out, right? And then the last one is going to be this electron transport chain and chemiosmosis, right? Together, that makes oxidative phosphorylation, right? And I apologize, it's kind of cut off here. Oxidative phosphorylation, all right? Remember that phosphorylation is just the sticking of a phosphate group onto something. In oxidative, right, we're going to be removing an H. So oxidative phosphorylation occurs in the mitochondria, just like the Krebs, and it is going to be responsible for making a lot of ATP. In this case, we're going to see like 34 ATP made for a single glucose, right? So together, we get about 38 ATP from glycolysis, Krebs, and oxidative phosphorylation, right? So one thing I want to point out is that you can see that if you look in this diagram again, we make ATP in different ways. So we make ATP by what's called substrate level phosphorylation in both glycolysis and the Krebs, and those both make about 2. And then we make a lot more ATP through what's called oxidative phosphorylation. So I just want to point out what those are. So, so small amounts of ATP are made by what's called substrate level phosphorylation, right? And so that's in glycolysis and the Krebs. So substrate level phosphorylation I don't know why, whatever, that's okay, is when an enzyme removes a phosphate group from something else. So let's call this protein A, whatever. It's going to remove a phosphate group from something else, and they're going to stick it on ADP. And that's how you make ATP. All right. So this is a transfer of a phosphate group from another protein, right? That is going to be very different than electron transport chain and oxidative phosphorylation, where you take free ADP and free floating inorganic phosphate, and together those are going to come together, right? Rather than removing a phosphate from one thing and sticking on another, this is free floating inorganic phosphate when we're dealing with oxidative phosphorylation. Okay, so I will do another screencast where I literally draw out every step of cellular respiration, pyruvate oxidation, Krebs, and the ETC, all right? So hold on for that. You will have an assignment, which I will post, so don't worry, where you are responsible for drawing out every stage as well. So get ready for it. Okay, so moving along. After glycolysis, where you cut glucose into pyruvate, the pyruvate goes into the Krebs cycle, you make some carbon dioxide, but in the meantime, what you're gonna be generating are these electron carrier molecules, these coenzymes called NADH and FADH2. And remember, these are their reduced forms, right? When they get fed into the electron transport chain, what you will see is that these carrier molecules get oxidized. So remember, when you have oxidized, you are going to basically remove an H. So if we look down here, what we see is, here. So what we're looking at here is NADH, and it is being converted to NAD+. So again, in my mind, we are going to remove an H, and that is going to be oxidizing it, right? In the same regard, we have FADH2 here, and it is going to go to FAD. We are going to remove an H, cross it out, so we're going to oxidize it, right? So during this process, we are oxidizing those high energy electron carriers, and what happens is that they're going to donate electrons to the electron transport chain. All right. So these NADH and FADH2 molecules get made in glycolysis, pyruvate oxidation, and Krebs. In the ETC, those are going to be oxidized, and the electrons are going to be given to the electron transport chain. These electrons get transported through the electron transport chain, right? So if I'm gonna try to make, I guess I'm gonna do it in red. Whoops. So these electrons are gonna be donated to the electron transport chain, either by NADH or FADH2. They are going to be moved like a conveyor belt, or a kind of like a manufacturing team. You take one person, takes it, gives it to the next, takes it, give it to the next, takes it, give it to the next, right? And when it does that, as those electrons are moved through the electron transport chain, what happens is that hydrogens get pumped into the intermembrane space. And that will create, it's moving, an electrochemical proton gradient, right? You are going to get a lot of H's hanging out in this area, which is the intermembrane space, right? So this is intermembrane space. And that is the space between the inner and outer membrane of a mitochondria, right? So we're going to generate this. Electrochemical gradient. And here, let me see if I can color that in. So the gradient is again be stored in this intermembrane space, which I'm kind of coloring over here in orange. And as the electrons, as these H's that we generated this electron transport chain, let me rephrase. Okay, so I'm gonna go back to the beginning here. So electrons move through this electron transport chain. At the same time, H's get pumped into the intermembrane space to create that gradient. The electrons are going to join with oxygen to create water, right? Which is an end product of cellular respiration. The H's are going to flow down their concentration gradients into the matrix, and at the same time, they're going to be going through this cool little lollipop-shaped protein called ATP synthase, and they're going to make energy in the form of ATP, right? So it's called oxidative phosphorylation because these carrier molecules are becoming oxidized, and it's called phosphorylation because we're really kind of joining ADP with its inorganic phosphates to form ATP, right? So the electron transport chain generates the gradient, and then ATP synthase creates the ATP. All right, I'm gonna show you this quick movie, so I'm gonna stop my share, and then let me go to the movie, and here, okay.
Molecular machine that works like a turbine to convert the energy stored in a proton gradient into chemical energy stored in the bond energy of ATP.
So it's this cool rotor that literally, it's like a little protein machine that moves.
The flow of protons down their electrochemical gradient drives a rotor that lies in the membrane. It is thought that protons flow through an entry open to one side of the membrane and bind to rotor subunits. Only protonated subunits can then rotate into the membrane, away from the static channel assembly.
Oh, you know what? I just realized that you guys weren't watching that. Oops. All right, we'll show that at class. Sorry. You know, I make some mistakes. All right, so remind me to do that, and we'll watch that in class sometime soon. Okay. So it's a very cool movie, but the whole point is that hydrogens are built up in their electrochemical gradient in the intermembrane space. They are going to slow down their concentration gradient through ATP synthase. And then we are going to see ADP and inorganic phosphate coming together to make ATP. Right. All right. So for every single glucose molecule that is broken through glycolysis, pyruvate oxidation, which we haven't really discussed, but we will, and the Krebs, and then oxidative phosphorylation, we are going to make about 38 ATPs for every single glucose molecule, right? So we get 2 ATPs from glycolysis, 2 ATPs from the Krebs cycle, and about 34 ATPs from oxidative phosphorylation, giving us about 38 ATPs, all right? And we're going to learn about all of the steps and all of these processes when we draw it out together. All right, so as we mentioned earlier, if you have oxygen, then you're going to go through exactly what we discussed in the mitochondria, which is aerobic respiration. And that is going to be oxidative phosphorylation, right? And that you need oxygen because oxygen is the final electron acceptor in the electron transport chain, right? However, sometimes we don't have oxygen, or there's some organisms, like especially some bacteria, that don't have even mitochondria, right? So this requires no oxygen. It occurs in the cytoplasm. It is the most ancient of all processes because every organism goes through glycolysis. And you get a little ATP out, right? And Mr. Anderson has good movies on this as well. So the process we're really talking about down here is this little part here, right? After glycolysis, this pyruvate can either go to the mitochondria if there's oxygen, or can stay in the cytoplasm if there's no oxygen. So if there's not enough oxygen, glycolysis can make a little bit of ATP. And we just talked, glycolysis can make 2 ATP, right, without going into the mitochondria. So if there's no oxygen, glycolysis can be coupled with a process called fermentation, and you can continue to make small amounts of small amounts of ATP, right? So pyruvate with oxygen goes into the mitochondria, and that's where we're going to see these other processes in oxidative phosphorylation. If there's no ATP, right? So no oxygen. If pyruvate is made through glycolysis and there's no oxygen, it is going to go through a process called fermentation. So fermentation takes no oxygen, and the whole purpose of fermentation is to regenerate your NAD+ so that you can do glycolysis again, right? And glycolysis can continue to give you a little bit of ATP, in this case, 2 ATP, and then you can do fermentation. Regenerate your NAD+, a little bit of energy out again. All right, so I'll draw that out for you again on another screencast. So one thing that I find super interesting is that we have been talking a lot about carbohydrates, and in particular, our glucose, C6H12O6, and how glucose gets broken down to give you a lot of ATP, right? It goes through glycolysis, Krebs cycle, and then oxidative phosphorylation. However, you don't eat just sugars, right? You also have a diet of fats and proteins, right? All of these things can be used as fuel for cellular respiration, but they enter the cycle at different points, right? So proteins, I know that some of you have heard of these high protein or high fat diets, right? That kind of, and what they do is instead of going through glycolysis early on, they enter the cycle at different points. So proteins can be made specifically into pyruvate through another process. Fats can be broken down, and you can get parts of either glycolysis or later in later points in the cycle. So I just want to point that out, that it's really important to see where your diet, I think it's just really interesting to note how these different foods are metabolized in your body, right? And one great example of something that we talked about earlier is that cellular respiration is controlled through your favorite allosteric activators and allosteric inhibitors. So remember, allosteric bind away from an enzyme's active site, right? They bind away from it, from an enzyme's active site. So an example here that is shown is that we have an enzyme called phosphofructokinase, right? Which here is right here. It is an enzyme involved in one of the steps of glycolysis, all right? And so phosphofructokinase can be stimulated by AMP, right? So it's a molecule that can bind and make that, and it acts as an allosteric activator, all right? And that activator can make phosphofructokinase work better, right? It can also be inhibited by a molecule, it can be inhibited by ATP, right? And so this is an example of feedback inhibition through an allosteric inhibitor. And so if there's enough ATP, ATP will bind to phosphofructokinase as a way of being like, yo, we've made enough. Stop working. We have plenty of ATP made, right? And that's a great example of feedback inhibition. All right, so that is where I'm going to stop. Remember that all these PowerPoints have lots of questions at the end that you should practice, and come to me with questions if you have them.